In this section, we just want to do an overview of drugs and their mechanisms, and the drugs that are used to treat bacterial infections. Remember from microbiology, we talked about different bacteria, and here just as a quick reminder, you have Helicobacter pylori in the stomach. In the respiratory tracts, you have Streptococcus pyogenes and Haemophilus influenza. So this is just a quick overview of the bacteria. Of course, listen to the pharmacy practice faculty and what they say you need to know. Just because a bacteria is associated with a certain area on this chart doesn't mean that in practice that's what the one you need to worry about. As far as mechanisms of action for drugs, we have those which the quinolones do DNA gyrase. RNA polymerase is the rifamycins, rifampin. Then we have those which have to do with protein synthesis. And remember, there's on the ribosomes for bacteria, there's a smaller sedimentation, the 30S, and there's a 50S. And some drugs will target the 30S, and others will bind to the 50S. Then we have drugs that affect the cell wall synthesis, basically the strength of it, and then you're going to have the things that they'll blow up, have lysis. And then folate has to do with antimetabolites in the folic acid cycle, and in some ways they deny the precursor for making DNA. So that's how that all groups together. Just to remind you about the different types of cells, bacterial cells, the gram-positive, the gram-negatives, they're also the acid-fast with the very thick cells. The distinction between the gram-positives is they have a thick murine cell wall, and inside of that is the plasma membrane, whereas for the gram-negatives, you have a periplasmic space that is in between two membranes. So looking again at the structures of the cell walls, it's not surprising that some drugs who affect cell wall synthesis or cell wall integrity will have different specificity for the gram-positive and the gram-negatives. And again, the idea is that if you weaken the cell membrane, then you get left with the plasma membranes, then the cells can lyse. And just to point out again that key feature is osmotic pressure, that any area that has many things dissolved, like a high salt content or high sugar content, it will draw water. And that then there's actually pressure, and we call that the osmotic pressure, that is pushing out from this. So that in general, if a bacteria does not have a way to keep water out, water will want to enter in. Ions will want to diffuse out, and that is it. Okay, so types of mechanisms. Again, you have the sulfonamides, which are going to be antimetabolites. They're going to be involved in the synthesis of thymidine ultimately, and if you inhibit these sulfonamides, you're going to not have thymidine, which is an essential building block for DNA. Then you're going to have, which we'll be starting out with, talking about the penicillins, cephalosporins, carbapenems, and monobactams. These will all inhibit the bacterial cell wall. Then you're also going to have vancomycin, polymyxins, and gramacetins also affect cell walls. The quinolones are going to be affecting an enzyme called topoisomerase, which is involved in the processing and replication of DNA. You're going to have the rifamycins, which will be inhibiting a DNA-dependent RNA polymerase, which is involved in the making of RNA. And then you're going to have the different drugs that will affect the ribosomes. These schemes then, encompasses all the mechanisms by which we inhibit bacterial walls, bacterial growth rather. We have bacteriostatic and bactericidal. Quick structural overview. Again, we're going to go into depth onto each of these. Again, you have for the DNA, the fluoroquinolones, and this is the quinolone structure. We have two rings together, and there's a fluorine attached. Rifamycin are a big macrocycle. And we'll also talk about metronidazole, which has to do with one electron transfers. There's also then those that weaken the cell walls. Again, penicillins, cephalosporins, all in those beta-lactams. Bacitracin, vancomycin, daptomycin, and polymyxins. These are all targeting the cell walls. Again, back to the osmotic pressure issue. The beta-lactams, you're going to, if it's not the parent, because it already has a built-up cell wall, the cells that come out of it that are growing, they're not going to have their cell walls formed correctly, and then you'll have a weakening in the membrane, and eventually it will burst. Those that are attacking the ribosomes, again, you're going to have this model where actually you have a 30S subunit to which actually the messenger RNA kind of gets bound, and then you have the 50S subunit through which will have a place where the transfer RNAs will bring in amino acid precursors, and they're going to match it up to the messenger RNA, and then you're going to have a growing polypeptide chain, and you're going to have, actually, see the little dot here, that's one amino acid, and you actually have the long chain gets transferred over to the new transfer RNA, and then things will kind of ratchet over. The transfer RNA with its missing the chain will then be removed, and then you'll have the growing chain have to move back over. And that process is called translocation. So different drugs will affect different things. It will stop the correct transfer RNAs from binding to the messenger RNA, or they'll stop the translocation step, and this is what we'll go through as we consider each drug class. And then for the dihydrofolate reductase inhibitors, we cannot make dihydrofolic acid from scratch, and this is the target. We need this because this ultimately supplies a methyl group that is needed to make uracil into thymidine to make that building block. So, but bacteria cannot absorb dihydrofolic acid, so they need to make it from scratch from dihydropteridine. And so one class of drugs, the sulfonamides, inhibit the dihydropterite synthases. And then following up on that, there's this folic acid cycle, and there's an enzyme. So this dihydrofolic acid is what the bacteria can make, and humans take it in from the outside. And then there's a step called dihydrofolate reductase, where this carbon double bond nitrogen gets reduced, and then part of that then actually takes the side chain from a serine, gets it incorporated into this reduced area, and it makes a ring here, and then it transfers this methylene group. Methylene is a carbon with two hydrogens on it, and here we have uridine, uracil, and it's going to transfer it here, and so there are cancer drugs that inhibit, like 5,4-uracil inhibits thymidylate synthase, but that is not used against bacteria. But the dihydrofolate reductase, we have inhibitors of that for bacteria, and that will then, if you can't make tetrahydrofolic acid, you can't grab this methyl group and it can't transfer it. So, and those are all the mechanisms.